Like every other system in the body, aging does have an effect on the integumentary system. So with age, we have wrinkles developing, dehydration of the skin, and cracking of the skin will occur. Sweat production will actually decrease as we age. We'll get a decrease in the number of functioning melanocytes, which really results in gray hair and atypical skin pigmentation. So maybe like aging spots would be an example of atypical skin pigmentation. Sub-Q fat is lost, and there's a general decrease in the thickness of the skin. And then nails would also become more brittle. Now, with aging, there's also an increased susceptibility to pressure ulcers, which we also call bed sores. Again, this is in part due to the general loss of subcutaneous fat and a general decrease in skin thickness. So with that in mind, pressure ulcers are caused by a constant deficiency of blood flow to tissues. So typically, the affected tissue is going to overlie a bony projection that has been subjected to prolonged exposure to an object or against an object like a bed or a cast or a splint. So essentially, bed sores could be as a result of staying in bed too long, staying in one position, having one part of their body being pressed up against a hard surface, like a bed or a cast or a splint, something like that, and not taking that pressure off. So if the pressure continues for too long, the tissue will die and we will see these pressure ulcers. A melanoma is a tumor of melanin-forming cells. Typically, they are malignant. So this is a term that essentially means it is cancerous and very dangerous. So malignant tumors are cancer tumors, whereas the opposite term, which is benign, a benign tumor is not going to be a dangerous tumor and it is not cancerous. So generally, melanomas are malignant. So we're going to briefly go over burns, which is another possible damage to the integumentary system. So a burn is going to be tissue damage to the integument, usually caused by excessive heat, electricity, radioactivity, or corrosive chemicals. Now, burns are also graded according to their severity. So depending on how much skin is damaged is also going to determine the grading. So here we have a first-degree burn. First-degree burn, it only involves the epidermis, and it's characterized by mild pain and erythema. Erythema essentially is just another term for redness. With first-degree burns, there are no blisters present. The skin functions generally remain intact. Immediate flushing with cold water may lessen the pain and damage caused by a first-degree burn. Now, generally, healing of a first-degree burn occurs in about 3 to 6 days, and it may be accompanied by flaking or peeling. And we know this all from sunburns. We've probably all had a lot of redness, some pain, and generally we get peeling of our skin as it heals. Second-degree burns, these destroy the epidermis and part of the dermis. In this type of burn, some of the skin functions are lost, and you see redness, blister formation, and edema. Now, edema is another term that we should know. This is the collection of watery fluid in tissues. With second-degree burns, we also do have a lot of pain resulting. Now in a blister, we can kind of see a blister on this upper picture as well. Up here, we can see a blister. This is where the epidermis separates from the dermis, and this is due to accumulation of fluid between them. So we actually kind of get a separation here along this line, just because there's a lot of fluid buildup in that area. Now, during a second-degree burn, the associated structures like hair follicles, sebaceous glands, and sweat glands usually are not injured. If there's no infection with a second-degree burn, they're usually going to heal without any skin grafting or anything in about 3 to 4 weeks, but you may have scarring after that. First and second-degree burns are generally collectively referred to as partial thickness burns. So that's for both first and second degree burns. And then here we have a third degree burn. This is also known as a full thickness burn. And this destroys the epidermis, the dermis fully, and often goes into the subcutaneous layer as well. In this type of burn, most skin functions are lost. These burns can actually vary in appearance from a really marbly white color to mahogany color to even like charred, dry wounds. So the appearance of them can vary quite a bit. There is quite a bit of edema. And the burned region is also numb because the sensory nerve endings in the area have been destroyed. Now, regeneration usually occurs very slowly with a full thickness burn, and a lot of granulation tissue will actually form before being covered by the epithelium. So because all of this tissue has been completely destroyed and lost all of its function, usually skin grafting can be required in order to promote healing and also help with minimal scarring. So if it's a large area with a third degree burn, they will usually help to help the tissue to heal and help to reduce scarring by actually taking skin from another area of the body and grafting it to the area where there was the burn. The seriousness of a burn is determined by its depth, like we just talked about, first, second, and third degree, but also to the extent of the area involved. A couple of other things that are taken into account to determine the seriousness of a burn would also be the person's age and general health. So according to the American Burn Association's classification of a burn injury, a major burn includes third degree burns over 10% of the body surface area. So major would be third degree burns over more than 10% of the body surface area, or second degree burns over 25% of body surface area or more, or also third degree burns on the face, hands, feet, or perineum. Perineum we can see is here. This is going to include the anal and urogenital regions. Now, quick means of estimating the surface area affected by a burn in an adult is called the rule of nines. So this is where we're going to get like the 10% or the 25% from. So this rule of nines is in this picture on the right here. So we're going to count 9% if both the anterior and posterior surfaces of the head and neck are affected. So for anterior head and neck, head and neck would be 4.5, and then posterior would be another 4.5. So pretty much the entire head and neck, front and back, anterior and posterior, would be 9% total. We would also count 9% for both the anterior and posterior surfaces of each upper limb. So we'd say here, anterior shoulders, arms, forearms, and hands being 9%, with 18% for both upper limbs. Now we would count 4 times 9% to a total of 36% for both anterior and posterior surfaces of the trunk, including the buttocks. So this is going to be anterior and posterior trunk, including the buttocks. We're also going to count 9% for the anterior and 9% for the posterior surfaces of each lower limb. This is going to be as far up as the buttocks. So this will be thighs is going to be 9%, and then legs and feet to be another 9%, for total of the lower limbs being 36%. And then we also count 1% for the perineum to equal 100% total. So this is just going to be our rule of nines to determine the surface area or estimated surface area affecting an adult with a burn. This is chapter 6. We're going to be covering the skeletal system. Specifically, we're going to be looking at bone tissue. So the skeletal system provides support by acting as a structural framework for the body, providing support for soft tissues, providing attachment points for the tendons of most skeletal muscles as well. It also protects the internal organs from injury, like the cranial bones protect the brain, and also like the rib cage protects the heart and the lungs. Most skeletal muscles attach to bones, and when they contract, they pull on bones to produce movement. Bone tissue makes up about 18% of the weight of the human body, and it stores several minerals, especially calcium and phosphorus. Now, bone tissue stores about 99% of the body's calcium, and on demand, the bone can release minerals into the blood to maintain homeostasis and distribute minerals to other parts of the body. Now, within certain bones, a connective tissue called red bone marrow produces red blood cells, white blood cells, and platelets in a process called hemopoiesis. So red bone marrow is going to produce red blood cells, white blood cells, and platelets. This process of creating these cells is called hemopoiesis here. Now, we also have yellow bone marrow. And yellow bone marrow consists mainly of adipose cells, which store triglycerides, and these triglycerides are a potential chemical energy storage. So essentially, these are the fat storage. Now, on a macroscopic level, the structure of a long bone, like the humerus, for example, has several parts. Now, a long bone, just the definition of a long bone, is one that is greater in length than it is in width. So first part we'll talk about is the diaphysis. This is going to be the long bone shaft that makes up the length of the bone. The epiphyses. These are going to be the proximal and distal ends of the bone, with the metaphyses being the region between the diaphysis and the epiphyses. Then we have the articular cartilage. Now this is a thin layer of hyaline cartilage covering the epiphyses where the bone forms an articulation with another bone. So the term articulation, for our purposes, it just means a joint, a joining part with another bone. So where a bone articulates with another bone, this is just where they join together. Next, we have the periosteum. This is going to be a tough connective tissue surrounding the diaphysis. The medullary cavity. This is also called the marrow cavity. This is going to be a hollow cylindrical space within the diaphysis. And lastly, the endosteum is a thin membrane that lines the medullary cavity. So as we can see on this picture here, kind of outlines all those parts we just talked about on the last slide. So here we can see the diaphysis is the longest portion of the long bone. And on the ends are going to be the proximal epiphysis and the distal epiphysis. So proximal is going to be closer to the majority part of the body. So here is going to be the proximal epiphyses for the humerus. Here is going to be the distal epiphyses for the humerus. Now in a growing bone, the metaphysis contains an epiphyseal growth plate. So within this metaphysis, we're going to see this epiphyseal growth plate. So this is going to be in a growing bone. This epiphyseal growth plate is a layer of hyaline cartilage that allows the diaphysis of the long bone to grow in length. Now it's at about, well, between 14 to 24 years old where the long bones stop growing. Now it's at this point, once those long bones stop growing, that the cartilage in the epiphyseal growth plate is replaced with bone. And once that metaphysis is bone, it results in a bony structure that we now call the epiphyseal line. So the epiphyseal growth plate is going to be cartilage, and once that growth of the long bone ceases, it turns into bone, and we now call it a epiphyseal line. On this picture, we can also see articular cartilage on the proximal epiphyses and also the articular cartilage on the distal epiphysis. Now, articular cartilage has several roles. One of them is to reduce friction. So it's going to be reducing friction at these articulation points. And it's also going to absorb shock at these freely movable points, or freely movable joints rather. So we're going to absorb shock. So it's going to be a physical absorption, a physical kind of protection barrier at the ends of these long bones. Now, just a side note, generally where there is cartilage, where it's not a joint, this cartilage is going to be covered by perichondrium. Now, perichondrium provides cartilage with a blood supply, and so it's very helpful to cover this cartilage to help the cells to grow, providing it with that nutrition filled blood supply. Articular cartilage, though, does not have perichondrium. So because it lacks that connective tissue, the articular cartilage lacks blood vessels, and so repair of damage is going to be very limited at these articular cartilage points. So damage repair is limited here. Because that perichondrium provides blood vessels. But again, that articular cartilage does not have perichondrium. Perichondrium is only going to be seen with cartilage that is not at freely movable joints. In this picture, we can also see that periosteum. So periosteum is a tough connective tissue sheath, and it is associated with blood supply that surrounds the bone surface wherever it's not covered by articular cartilage. Periosteum has an outer fibrous layer of dense irregular connective tissue, and it also has this inner osteogenic layer that consists of cells. And some of these cells within the periosteum allow the bone to grow in thickness, but not in length. So we're going to say here these. So to grow in thickness. On this picture, we can also see that medullary cavity, this cavity inside the diaphysis of a long bone. And within this medullary cavity, it contains yellow bone marrow. There's also going to be a lot of blood vessels within this cavity too, like this nutrient artery. Now this cavity is going to be really important because it minimizes the weight of the bone by reducing the dense bony material where it's least needed. So a long bone's kind of tubular design provides maximum strength with minimum weight. And that medullary cavity helps a lot with reducing the overall weight of the bone. And the last part we're going to talk about here is the endosteum here. So this is going to be this really thin membrane that lines the medullary cavity. Now it does contain a single layer of bone-forming cells and a small amount of connective tissue in the endosteum. So that was looking at the macroscopic level of bone, kind of looking at the parts of bones that we can see with our naked eye. So now we're going to briefly discuss the microscopic levels, and looking at bone on the microscopic level, like other tissues, bone contains an abundant extracellular matrix that surrounds these widely separated cells. Now, the extracellular matrix of bone contains various different things, about 15% water, 30% collagen fibers, and about 55% crystallized mineral salts. Now, you don't specifically have to remember these numbers or anything, but just note that there's a lot of crystallized mineral salts within osseous tissue or within bone. Now, the most abundant mineral salt is going to be calcium phosphate. Calcium phosphate going to be that most abundant mineral salt, and it combines with other less abundant minerals like things like calcium hydroxide or calcium carbonate, and these combined mineral salts are deposited in the framework by collagen fibers of the extracellular matrix, and then they crystallize in a process that is called calcification. Now, this calcification process is initiated by bone building cells, which are called osteoblasts. And whenever we talk about cells, if we look at this suffix blasts, blasts means creating or building. And on the other hand, when we see this suffix, clasts, this is going to mean breaking or destroying. So we're just going to look at kind of these four main cells that we see on the microscopic level of bones. So first off, we have these osteoprogenitor cells. These are known kind of as the bone ancestor cells or the predecessor cells. These are going to be bone stem cells that are able to differentiate into other types of cells. Next, we have these osteoblasts. Again, these are going to be bone building cells, and these are cells that secrete bone matrix. Osteocytes are just mature bone cells. They don't build or destroy any longer. They're just kind of the solid cells within the matrix. And we'll look at these in a bit more detail a little bit later. Then we have osteoclasts. These are going to be the bone breakers. They're going to be essentially the cells that remodel bones, cause the bone to release calcium into the bloodstream. So if we are low in calcium in our blood, osteoclasts will be initiated to break down some of our bone in order to release some calcium into the blood. But they are very useful in remodeling, destroying some parts of bones, and then the osteoblasts can come and build bone in other parts. So this is what those cells look like. So here we have that osteoprogenitor cell. These are unspecialized stem cells. And because they are a type of stem cell, these are going to be the only bone cell to undergo division and proliferation. So these are going to be the proliferating cells, those dividing cells. So when osteoprogenitor cells divide or proliferate, it results in cells that develop into osteoblasts. Now these osteoprogenitor cells are found along the inner portion of the periosteum, also in the endosteum, and in the canals within bone that contain blood vessels. So these are going to be really important ancestor cells. Then we have osteoblasts. So these are going to be those bone building cells that synthesize and secrete collagen fibers. So they create it and they also secrete it. And they also create other organic components needed to build extracellular matrix of bone tissue. And another role is that they initiate bone calcification. So essentially the hardening of the bone matrix, that calcification, these osteoblasts will initiate that. Now, an interesting thing is that the osteoblasts will essentially secrete so much collagen fibers and extracellular matrix and kind of surround themselves with the extracellular matrix that they become trapped in their own secretions. And once they become trapped, they kind of turn off all of their functions and they become osteocytes. So osteocytes, these are going to be mature bone cells, and these are going to be the main bone cells within bone tissue. They maintain their own daily metabolism, like the exchange of nutrients and waste with blood, but they don't really have any other functions other than that. They don't create bone, they don't destroy bone, they don't undergo cell division or anything like that. They just kind of maintain their own day-to-day life. These are just going to be the main cells that make up your bones. Then over here on the right, we have our final cell type, which are osteoclasts. Now these are going to be really huge cells compared to the other three, and these are derived from the fusion of as many as 50 monocytes, which are a type of white blood cell. So these are going to be from that white blood cell lineage. So it's kind of a little separate here. We can kind of see this division line because osteoclasts are not built from osteocytes or anything like that. So they're from their own lineage completely. These cells are going to be concentrated in the endosteum. Now on the side of the cell, on the side of the osteoclast that faces the bone surface, the plasma membrane is going to be deeply folded into this ruffled border. So you kind of imagine that we have a long bone kind of here, and so the ruffled border is going to face the side of the bone. Now from this ruffled border, this is where the cell releases these powerful lysozymal enzymes and acids that digest the protein and mineral components of the bone matrix. So lysozymal enzymes and acids are going to come from that ruffled border and kind of break down and digest that protein and mineral within the bone. Now, the breakdown of the bone extracellular matrix, we call this reabsorption. This is really just a part of normal development. It's part of normal maintenance and repair of bone. So it's not an abnormal process. The breakdown and rebuilding of bone is a very, very normal process in development. So there's a couple different types of bone tissue, mainly compact bone and spongy bone. So compact bone tissue contains very few spaces, and it's the strongest form of bone that makes up the bulk of the diaphysis of long bones. Compact bone is really good for providing protection and support, and it resists the stresses that are produced by weight and movement. So it's going to be your very much structural support bone type. Now spongy bone, which is also referred to as trabecular bone tissue, and we'll see why in a second, but spongy bone is always located on the interior of bone. So we can see spongy bone over here is always going to be protected by compact bone on the outside. So even here on the epiphysis that we can see, we're going to have spongy bone on the inside here, but we're always going to have that layer of compact bone around the outside. In the diaphysis here, we can kind of see it's a little bit thicker layer of compact bone here. But yeah, we're always going to have spongy bone protected by compact bone. So here in this picture, we can see our compact bone and our spongy bone. So we're going to have that spongy bone on the interior with that compact bone protecting it on the outside. So first we'll talk about compact bone. This is composed of repeating structural units that are called osteons, which are also called haversian systems, the entire osteon. And each osteon consists of concentric lamellae that are arranged around a central canal. Central canal is also called a haversian canal. And kind of like resembling the rings of a tree, the concentric lamellae are going to be these circular plates of mineralized extracellular matrix that increase in diameter as they get further away from the central canal. These concentric lamellae also surround a network of blood vessels and usually a nerve as well that's located within that central canal. Now these are going to be tube-like units that form a series of parallel cylinders that run parallel to the axis of the bone. So if we have a long bone that's kind of running this way, sorry, my bad picture of a bone, but so if the long bone is running kind of this way, top to bottom, these osteons with their concentric lamellae are also going to run parallel to the length of the entire bone. Now between the concentric lamellae are going to be these small spaces called lacunae, or a singular lacuna. Now these lacunae is a term that actually means little lakes. And so these little lakes, I'll put this here, little lakes, these lacunae will actually contain osteocytes within them. So we can see an osteocyte here with the lacuna surrounding the osteocyte. This is just a blown up version of what we can see here, kind of all around in these concentric lamellae. Now radiating in all directions from the lacunae are going to be little tiny canaliculi. Canaliculi are going to be these really small channels which are filled with extracellular fluid. Now think of it if you were to put lots of circles together, so a circle here, and we'll have a circle here and a circle here. If you put all these circles together, it's not going to create a completely filled surface, right? You're going to have lots of spaces in between these circles that isn't filled in. So that's where we get these interstitial lamellae. You can kind of see, if I raise this a bit, you can kind of see an interstitial lamellae here, in between those concentric lamellae. So this is going to be that area between neighboring osteons that contains lamellae that we call interstitial lamellae. These interstitial lamellae also have lacunae with osteocytes and canaliculi. Interstitial lamellae are actually fragments of older osteons that have been partially destroyed during bone remodeling or growth. So they're kind of just filling in those spaces, but they used to be full osteons. Well, what about spongy bone? So spongy bone does not contain osteons, but it does consist of lamellae that are arranged in irregular patterns of thin columns that we call trabeculae. Now between trabeculae are going to be spaces that are not visible to the naked eye, but these are spaces that are filled with red bone marrow. This red bone marrow is producing red blood cells. Red blood cells and also white blood cells. Now these spaces have a really good supply of nourishment from blood vessels to produce or provide nourishment to the osteocytes. So we can see osteocytes within those trabeculae. You can also see these interstitial lamellae here. They're not as organized as we saw with the osteons, but we still have these lamellae with lacuna surrounding the osteocytes and also with the canaliculi too. Now, spongy bone is a little bit different from compact bone in two aspects. So the first one is that spongy bone is very light, and this is going to reduce the overall weight of the bone. So it's very lightweight. Compact bone is much heavier in comparison. Second difference we see with spongy bone is that the trabeculae of spongy bone support and protect red bone marrow. So we don't see that red bone marrow in compact bone. So just kind of these two differences, or major differences, between spongy bone and compact bone. The spongy bone in the hip bones, the ribs, the sternum, and vertebrae, as well as the proximal ends of the humerus and femur, are going to be the only sites where red bone marrow is stored, and so these are going to be the main sites where hemopoiesis occurs in adults. So again, this is going to be the hip bones, the ribs, the sternum, the vertebrae, and the proximal ends of the humerus and the femur. So again, these are going to be the only places where red bone marrow is stored within the spongy bone, and this is where we're going to have hemopoiesis occur. Okay, let's talk about how bone is formed. So the process by which bone forms is called ossification, also known as osteogenesis. Now, bone formation occurs in 4 principal situations. So this is going to be the initial formation of bones in an embryo and a fetus. Secondly, the growth of bones during infancy, childhood, and adolescence up until the adult sizes of bones is reached. Also, bone formation occurs when bone remodels. So this is going to be the replacement of old bone by new bone throughout life. And then lastly, the repair of fractures throughout life. So first, we're going to be looking at bone formation in the embryo. So the embryonic skeleton is initially made of mesenchymal cells in the general shape of a skeleton, and this is the site where cartilage formation and ossification occurs, usually during about the 6th week of embryonic development. So this is where we're going to have that cartilage formation and ossification, that creation of bone, happen in the 6th week. Now there are 2 patterns of bone formation, both which involve the replacement of pre-existing connective tissue with bone. These don't lead to differences in the structure of mature bones, but these are going to be just simply different methods of bone development. So the first type of ossification is going to be intramembranous ossification. This is where bone forms directly within mesenchyme, and it's arranged in sheet-like layers that resemble membranes. And then we're going to talk about endochondral ossification just a little bit later. So here we have intramembranous ossification, and this is the simpler of the 2 methods of bone formation. So the flat bones of the skull, so here we can see those, the flat bones of the skull, also most of the facial bones, the mandible as well, the lower jawbone, and the medial part of the clavicle are all formed in this way with intramembranous ossification. So here we have the 4 steps of intramembranous ossification. So first off, we have number 1, the development of the ossification center. So at the site where the bones will develop, there's going to be special or very specific chemical messages that cause the cells of the mesenchyme to cluster together and differentiate first into osteoprogenitor cells and then into osteoblasts. So the site of this cluster is called an ossification center. And this is where the osteoblasts secrete the organic extracellular matrix of bone until they are surrounded by it. So again, we have that site of clustering of osteoprogenitor cells. So they cluster at the ossification center, and the osteoprogenitor cells develop into osteoblasts, and the osteoblasts will secrete extracellular matrix of bone until they are completely surrounded by that extracellular matrix. Next, we have step 2, which is calcification. So this is where the secretion of the extracellular matrix stops, and then the cells, which are now going to be osteocytes, they lie in the lacunae, and they extend their small little extensions into the canaliculi, and these are going to be radiating in all directions. Calcium and other mineral salts are going to be deposited within a few days, and the extracellular matrix hardens or calcifies. So that is the calcification step. Step 3 is the formation of trabeculae. So as the bone extracellular matrix forms, it develops into trabeculae that fuse with one another to form spongy bone around the network of blood vessels in the tissue. So here we have those spongy bone trabeculae forming around the blood vessels. Now the connective tissue associated with blood vessels in the trabeculae is going to differentiate into red bone marrow. So this is where that red bone marrow is formed. Then we have step 4, which is the development of the periosteum. Now this almost happens at the same time with the step 3, the formation of trabeculae, but at this time the mesenchyme up here, we can see that also the mesenchyme is condensing at the periphery of the bone, and it develops into periosteum. Eventually, there is a thin layer of compact bone tissue that replaces the surface layers of the spongy bone. And then we have the spongy bone kind of remaining just in the center. Now, even after this type of bone formation is completed, a lot of this bone will be remodeled, so it'll be destroyed and then reformed. And this remodeling all happens as the bone is transformed into its adult size and shape. And then we have endochondral ossification. So in this type of bone formation, the bone forms with hyaline cartilage that develops from mesenchyme. So essentially, cartilage is formed first in the correct shapes and everything, and then that is going to be replaced with bone. Now, most bones in the body are formed in this way, but it is best observed in a long bone. So we're going to kind of use a long bone here as an example for endochondral ossification. Now, essentially, there are 6 steps for endochondral ossification. Our first one here is number 1, this development of a cartilage model. So at the site where the bone is going to form, specific chemical messages will cause the cells in the mesenchyme to crowd together in the general shape of the future bone. So this part is pretty well the same between intramembranous ossification and endochondral ossification. But then we kind of differ because those cells kind of cluster together and then they develop into chondroblasts. So these are going to be cartilage creating cell. Chondro meaning cartilage, blast meaning creating. Now these chondroblasts will secrete cartilage extracellular matrix, producing a cartilage model. Now this cartilage model consists of hyaline cartilage. There's also going to be a covering of perichondrium that develops around this entire cartilage model. Step 2 is the growth of the cartilage model. So once these chondroblasts are covered in the extracellular matrix that they secrete, they are now called chondrocytes. This is very similar to osteoblasts turning into osteocytes, but these are cartilage cells. So they are chondroblasts developing into chondrocytes. The chondrocytes actually will continually divide and produce more extracellular matrix, adding to the length of the cartilage model. Now, as the cartilage model grows in size, the chondrocytes in the middle will increase in size, or hypertrophy. It's a term we also should know. So these cells themselves will grow in size, the cells will hypertrophy, and the surrounding extracellular matrix in the middle begins to calcify. Then in step 3, we have the development of the primary ossification center. So this is important. Primary ossification centers proceed inward from the external surface of the bone. So here we go, primary. This proceeds inward from the external surface of bone. We'll just put external bone here. So during this stage, we have a nutrient artery that is going to penetrate the perichondrium and also the calcifying cartilage model through a nutrient foramen. And this is going to stimulate the osteoprogenitor cells in the perichondrium to differentiate into osteoblasts. Now, once the perichondrium starts to form bone, then it's called periosteum. Now, near the middle of the model, the capillaries will grow into the disintegrating calcified cartilage, and this induces the growth of the primary ossification center. And this is going to be the region where bone tissue is will replace most of the cartilage. So primary ossification center, we're going to be replacing cartilage with bone, starting from the outside and going internally, proceeding inward from the external bone. Step 4 is going to be the development of the medullary cavity. So as the primary ossification center grows towards the ends of the bone, the osteoclasts break down some of the newly formed spongy bone trabeculae. Now, this activity leaves a cavity, the medullary cavity, and this is going to be within the diaphysis. Eventually, most of the wall of the diaphysis is replaced with compact bone. Step 5 is the development of the secondary ossification centers. Now, in contrast to the primary ossification, secondary ossification proceeds outward from the center of the epiphysis towards the outer surface of bone. So this is going to proceed outward. Now, when branches of the epiphyseal artery enter into the epiphysis, secondary ossification centers are going to develop. Now, usually this happens around the time of birth. Now, this is pretty similar to the primary ossification, though the spongy bone remains in the interior of the epiphysis, and there's going to be no medullary cavity found here. Then the last step of endochondral ossification is the formation of articular cartilage and the epiphyseal or growth plate. So the hyaline cartilage that covers the epiphysis becomes articular cartilage. And prior to adulthood, hyaline cartilage remains between the diaphysis and the epiphysis as the epiphyseal growth plate. Now this is going to be that region responsible for the lengthwise growth of long bones, or the getting longer. So those are pretty much the 6 steps of the endochondral ossification. We're going to have that development of a cartilage model, which is going to be replaced with bone, first at the primary ossification center and then from the secondary ossification center to replace that entire cartilage model with bone. Now, the growth in length of long bones involves the following 2 major events. So first one is going to be the interstitial growth of cartilage on the epiphyseal side of the epiphyseal plate. And then our second big event in order to increase the length of long bones is going to be the replacement of cartilage on the diaphyseal side of the epiphyseal plate with bone. And this happens by endochondral ossification. So this is on the diaphyseal side. So essentially, the cartilage is going to be growing in one direction and the bone is going to be replacing it, kind of following behind. So this initial growth, this is going to be interstitial growth. Now recall that the interstitial growth involves this continual cell division of chondrocytes and further secretion of cartilage extracellular matrix. So here on this radiograph on the left, we can see the epiphyseal plate. So here we can see this radiograph, this epiphyseal plate here, and below it is going to be a really blown up magnified version. So here on this growth plate, we can actually see on the radiograph, it appears as a dark band between these white calcified areas on the X-ray. And this is because bones will look white, but anything else, like cartilage, is going to appear black or darker. Now there are zones in the epiphyseal plate. There's going to be resting cartilage down here. This is on the epiphyseal side. And these do not contribute to bone growth. Essentially, these are going to be anchoring to the epiphyseal, or anchoring the epiphyseal plate to the epiphysis of the bone. Next is the zone of the proliferating cartilage here. So this zone is going to include chondrocytes that undergo interstitial growth as they divide and secrete extracellular matrix. The zone of hypertrophic cartilage, this consists of really large maturing chondrocytes that are arranged in columns. And you can pretty well see these here. You can see these columns of large chondrocytes. Then we have the zone of calcified cartilage here. And this consists mainly of dead chondrocytes because the extracellular matrix around them has calcified. The osteoclasts will dissolve the calcified cartilage, and the osteoblasts and capillaries from the diaphysis will invade the area and replace the calcified cartilage by endochondral ossification. So we're going to replace it with bone. So here further on we have bone. So bone grows in this way by the proliferation of chondrocytes, which then die and are replaced by osteocytes. This is how bone grows in length. So let's discuss what happens if those bones break. So a fracture is any break in a bone, and there are many different types of fractures that can occur. Now the healing process involves 3 different phases in 4 steps. First off, we have the reactive phase. This is going to be an early inflammatory phase. The reparative phase is broken up into 2 different steps. This is where the fibrocartilaginous callus formation occurs. And then after a bony callus is formed. Then we have a bone remodeling phase. This is going to be the final phase where the bony callus is removed and a healed fracture line may remain. So we can see these phases and their steps here in this picture. So during the reactive phase, this is when blood vessels crossing the fracture line are broken as blood leaks from the torn ends of the blood vessels. There's usually a mass of blood that's clotted. It forms around the site of the fracture. Now this mass of blood, which is called a fracture hematoma, usually forms about 6 to 8 hours following the injury. Swelling and inflammation occur. And these occur in response to the dead bone cells in the area. And this is due to the blood stopping at the site where the fracture hematoma forms. So blood cannot cross the fracture, and so there's going to be a lot of cells that die because they're unable to get nutrients. Now, this swelling and inflammation causes an influx of phagocytes. Specifically, these are going to be neutrophils and macrophages that come into the area. And during this phase, the osteoclasts will begin to remove the dead and damaged tissue. Now, this stage may last several weeks. Next, we have the reparative phase, which is broken up into 2 steps. This is first part is going to be the fibrocartilaginous callus formation step. And this is when the blood vessels grow into the fracture hematoma, and phagocytes begin to clean up those dead bone cells. This is where we have fibroblasts from the periosteum will invade the fracture and start to form collagen fibers. Also, we have chondroblasts in the region, which are going to form fibrocartilage, and this leads to the development of this fibrocartilaginous callus. This is also just a soft callus. This is a mass of repair tissue consisting of collagen fibers and cartilage that bridges the broken ends of the bone. Now, formation of the fibrocartilaginous callus takes about 3 weeks to fully form this fibrocartilaginous callus. Now, the areas to well-vascularized healthy bone tissue, or in these areas, we have osteoprogenitor cells develop into osteoblasts, and they'll begin to develop spongy bone trabeculae. Now, the trabeculae will join living and dead portions of the original bone fragment, and then in time, the fibrocartilage is converted into spongy bone, and it's at this point that the callus is then referred to as a bony callus, and it's now going to be a very hard, bony callus. Now, this bony callus lasts about 3 to 4 months. Now, the final phase of fracture repair is bone remodeling of the callus. So dead portions of the original bone are gradually reabsorbed by osteoclasts, and compact bone replaces spongy bone around the periphery of the fracture. That's an important part. So around this area here, this is where we have compact bone replaces that spongy bone. This point we also have a healed fracture, then it's just going to be a line instead of any callus. Fractures, just like burns, are named according to their severity, the shape or position of the fracture line, or sometimes even just the physician who first named them. Now, there are lots of different ways that a bone can break, but we're just going to cover 3 common types. So the first one is an open or a compound fracture. These fractures occur when the broken ends of the bone actually protrude through the skin. So we can see that here. There we have comminuted. These fractures are when the bone is splintered, crushed, or broken into pieces at the site of the impact. And then we're going to see smaller bone fragments between the 2 main pieces. So again, splintered, crushed, or broken into pieces at the site of the impact. And then here we have a green stick fracture. Now, green stick fractures are partial fractures in which one side of the bone is broken. You can see here, this side of the bone is broken, and then the other side of the bone bends. Now, this is going to be similar to the way that a green twig breaks on one side while the other side stays whole but bends. Usually this occurs only in children, and this is because their bones are not fully ossified and they contain more organic material than inorganic material. So it's not a break all the way through the bone. Now, bone is the body's main calcium reservoir, storing 99% of the body's total calcium. Now, one way to maintain the level of calcium in blood is to control the rates of calcium reabsorption from bone into blood. and also to control the rates of calcium deposition from blood back into bone. Now, calcium ions are needed for nerve and muscle function, as well as blood clotting and several enzymatic reactions. So this is what calcium is needed for. Nerve and muscle function, blood clotting, and enzymatic reactions. These are going to be the main places where we need calcium. Now, calcium exchange is regulated by hormones, the most important of which is going to be the parathyroid hormone, often abbreviated PTH. And this is going to be secreted by the parathyroid gland. Now, this hormone increases blood calcium levels. So, increasing the blood calcium. Also, the parathyroid hormone secretion operates by a negative feedback mechanism. Parathyroid hormone also acts on the kidneys to decrease the loss of calcium in the urine so that more calcium is retained in the blood. Parathyroid hormone also stimulates the formation of calcitriol. Again, this is the active form of vitamin D. And calcitriol is a hormone that promotes absorption of calcium from foods in the gastrointestinal tract, and it moves that calcium into the blood. So both of these actions also help to elevate these blood calcium levels. Parathyroid hormone acting on the kidneys, to decrease calcium loss in the urine. And also the stimulating the formation of calcitriol. So these are both major mechanisms that help to elevate blood calcium levels. So release of calcium from the bone matrix and retention of calcium by the kidneys are two main ways that the blood calcium levels can be increased. So here we have a negative feedback mechanism that occurs when we have blood calcium levels decreased. It's going to be detected by the parathyroid glands. And this is going to cause an increase in production of cyclic AMP, which turns on the parathyroid hormone gene. And turning this gene on increases the release of parathyroid hormone. Now, this parathyroid hormone is going to have two major effects. It's going to act on osteoclasts to break down bone matrix and release that calcium into the blood. So we're going to increase bone reabsorption. So that's going to be about breaking down of bone. And the parathyroid hormone is also going to act on the kidneys. So the kidneys are going to retain more calcium in the blood and produce calcitriol. So these responses are generally all going to work together to increase the blood calcium level, returning us back to homeostasis. So just like we've discussed before, aging does also have an effect on bone tissue. One major one here is osteoporosis. So in osteoporosis, bone reabsorption outpaces bone formation. So we're going to say that the osteoclasts, these are working more. They're going to have an increased work than osteoblasts. So what this means is that bone is going to be broken down faster than it is created. And so then bone mass decreases. And that will be the end of chapter 6.